Good day. I will be presenting my model of the cell membrane. Before showing the cell membrane, I would like to present first my model of the glycerophospholipid. The violet binder ring represents the glycerol. Being the backbone of the molecule, the binder ring is able to connect both the phosphate group and the fatty acid chain. On the topmost part of the structure, we can see a violet circle, which is the lock of the binder ring. This represents the phosphate group, which is the hydrophilic head of the glycerophospholipid. I did not add any more items to represent the phosphate group, since the lock of the hoop is easily identifiable and fits the structure it represents. The bottom area, as well as the hydrophobic tail of the molecule, is the fatty acid chain represented by a remodeled paperclip. I reformed the paperclip this way in order to fit the structure of the fatty acid as presented in the resource materials. As a whole, these three molecules make up the glycerophospholipid. It is an amphiphilic molecule having both hydrophobic and hydrophilic ends. This is a characteristic of the cell membrane that allows it to accommodate both polar and nonpolar molecules. Here is my cell membrane model. This orange part here is the extracellular environment, while the pink part here is the intracellular environment. The cell membrane is a phospholipid bilayer as seen by the two layers of the phospholipid molecules explained earlier. This one here is one layer, and at the bottom is another layer. This bilayer is structured in such a way that the hydrophobic tails meet in the middle, while the hydrophilic heads are facing both inwards and outwards the cell. Now let's explore the other molecules found in the membrane aside from the glycerophospholipid. First, the proteins. There are 4 proteins found in this model: 1, 2, 3, and 4. The first 3 are what we call integral proteins, which are embedded in a membrane, which is why these structures are sandwiched between glycerophospholipids. This protein right here is a peripheral protein. This protein is not embedded inside the membrane and is attached onto an integral protein. The protein represented by a clothespin is a transport protein. It is a kind of an integral transmembrane protein that facilitates inactive transport. The violet ends of the clothespin represent the hydrophilic parts of the protein. The remaining green part represents the hydrophobic areas of the protein. Since the clothespin opens and closes, it may show the process of active transport through allowing certain molecules to pass through. I will explain on this further later. This pearl hair clip represents another integral protein. Attached to it is a chain of staple wires. A set of staple wires can also be found here, attached to the glycerophospholipid. These chains of staple wires represent carbohydrates. This molecule is called glycolipid, while this molecule is called a glycoprotein because of the attachments of the carbohydrates. Over here and here, we can see a cluster of staple wires which represents cholesterol, linking together glycerophospholipids in order to help regulate the fluidity and movement of the molecules in the membrane. The cluster of staple wires restricts the movement of the binder rings to a certain extent. For example, here, it cannot go further than this, and here, it only stays up to this extent. One of the characteristics of a cell membrane is that it is semi-permeable. Because of this, only certain molecules are allowed to pass through the membrane, and the others can only enter and exit the cell through the help of proteins. For example, this item, representing a small molecule, let's say a water molecule, is able to pass through the membrane. It is not as quick as gases, but still, the membrane allows it to enter freely. Meanwhile, this item representing a large molecule, for example an amino acid molecule, requires the assistance of a transport protein in order to pass through. Earlier, we stated that this clothespin represents a transport protein. In the process that this molecule needs to enter the cell, it requires the help of this protein, for example like here, and then it will be able to go here and enter the cell. Cell membranes are fluid. In moving the plastic underneath the components, just like this one, the items on top also moves. It shows that the cell membrane is not entirely rigid, but rather it exhibits fluidity. Also, it is fluid in the sense that proteins are able to move laterally. For example, this protein can move over here, or this protein can move over here. Cell membranes are mosaic, the whole mix of the different items representing different components of the cell membrane exhibits mosaicity. The binder ring, the paper clip, the dropper, the clothespin, the cluster of staple wires are all individual pieces that make up the whole, which is the cell membrane. Thank you.